Hi, everyone. Welcome to OsteoBites, and thanks so much for joining us today. My name is Christina Iptoma, and I am mom to Osteo Angel Dillon and Director of Scientific Programs at MIB Agents. And today on OsteoBites, we are joined by Dr. Yingqi Hua from Shanghai General Hospital, Shanghai Jiao Tong University School of Medicine, and he will be discussing his work on molecular subtyping of osteosarcoma, ecDNA, and more. Thanks so much, Dr. Hua, for joining us on OsteoBites today, especially at this very early hour for you. Everyone, please feel free to submit questions during the presentation through the Q&A in Zoom, and we will share those with Dr. Hua after his presentation. A little bit more about our guest today. Dr. Hua has been engaged in basic and clinical research of malignant bone tumors for more than 10 years, focusing on the individualized comprehensive treatment of advanced bone tumors, and he's committed to transforming research results through clinical research. His research interests include the multi-omics study of osteosarcoma, where multi-omics analysis identifies osteosarcoma subtypes with distinct prognosis, indicating a stratified treatment. Also, the epigenetic study of bone tumors, the function of histone mutation and the function of different histone methylation modification. Also, drug screens, screening sensitive drugs by patient-derived xenograft and exploring the mechanism of drug action. And lastly, clinical trial of advanced stage sarcomas based on precision medicine. So we are thrilled to have you. Thanks so much for joining us today, Dr. Hua. Just a few announcements before we get started. There is still time to register for our Factor Conference in Salt Lake City, June 26th to 28th. I will put a link in the chat to more information about how to register. And we have a discounted conference room block at the Grand America Hotel. There are still a few rooms available, but you have to book by June 6th to take advantage of that discounted rate as the conference block is closing soon. Not only do we have some of the best and brightest in the scientific community coming together to collaborate on osteosarcoma at Factor, but it really is a unique conference so that it brings together the scientific, patient, advocate, and funder communities and gathers everyone in place to work on solutions together. Our scientific program is available online. I'll put a link in the chat where you can find it. We have 7 scientific panels, as well as our very popular patient panel. We have 2 scientific breakout sessions that are great opportunities to come to Salt Lake City and engage in the conversation at the conference. One of the breakouts will be led by Sark, and that will be focusing on mentorship and coaching for young investigators. And I did want to highlight the second breakout session that may be of particular interest to our OsteoBites audience today, and that is an interactive session that is going to be led by the Cure OS working group. And that group is focused on creating a blueprint for translating single-cell discoveries into better osteosarcoma treatments. And that group will review evolving recommendations on using single-cell technologies to study intratumoral heterogeneity and the tumor microenvironment, and discuss strategies for translating these insights into personalized treatments. So it's going to be an interactive session. I know that they're planning on having some kind of roundtables during the group to really discuss and kind of come up with some deliverables for moving forward. So that should really be a great breakout session. And I'll put some more information in the chat about all of this. And otherwise, Dr. Hua, I will hand it over to you to start your presentation.
Hello, everyone. It's a great opportunity to have this meeting and discuss the disease we are interested in. Let me just firstly show my slides. Let's see, okay. Is
that
it
looks great.
Available?
Yep, that looks good.
Okay. I'm from Shanghai General Hospital, Shanghai Jiao Tong University, bone tumor lab. And today I'm talking about some of my work from our lab. And it's mainly about what we are doing in the molecular subtyping of osteosarcoma, which I think this is that we have this group to communicate is very wonderful. So this is a disease that we are all interested in, and however, we cannot further elevate the outcome for this patient. And this is partly because that we need to learn more about the genomic backgrounds of osteosarcoma. One of the thing that has not been fully studied or characterized is the genome event, which is Chromothripsis, which is very common in osteosarcoma. As we can see, the high frequency of all cancers is these two sarcoma type. One is liposarcoma, one is osteosarcoma. And this leads to its abnormalities in the osteosarcoma, for example, Chromothripsis and the contagious and the extensive copy number variation and highly genomic instability. Actually, more than 10 years ago, people found this in osteosarcoma, that the genome is shattered and rearranged. And during that, many genes were lost, and some of them is highly amplified. So this is the event characterized as Chromothripsis. However, the mechanism of Chromothripsis is not fully understood. There's some paper coming these years trying to understand how it's a event during the mitosis, how the genome is ruptured and then combined again. However, there's still a lot of things that we are not aware of. And there's also some attempts trying to target these highly instability cancer. For example, there's some drug trying to target the KIF18A, which might be synthetic lethal to this Chromothripsis cancers. This is a fairly important work that has been published early this year. And I think there's a lot of work that has been done from this group. It has described this common event, Chromothripsis, in a subclonal event in bone soft tissue sarcoma, especially osteosarcoma. About half of the cases presented with a lost translocation amplified type, which could be mediated by p15-3 disruption. And there's genome-wide loss of heterozygosity may serve as a prognostic biomarker in high-grade osteosarcoma. I think everyone has read this. There's a lot of information to describe this genomic event. But there's still a lot of things that we need to study. For example, to detect or to diagnose with Chromothripsis, there's different methods and different sequencing approaches, but there's some difference between studies. And the most important question is, how can we target, or there's some kind of drug, maybe the synthetic lethal, from the synthetic lethal point of view, or to target the genomic instability. And the most important thing is, I think this, because the genomic is shattered in a random way, and then it causes a lot the heterogeneity that we can see in the clinic. Some of the patients has shown a very good prognosis, and some of them is poorly has very poor outcome, even though we have the same pathological feature. They all look the same under microscope, but actually because of this Chromothripsis event, their genome is varied a lot. So this is the work we've been done through about 10 years, and then we're trying to use multi-omic approach to have this profile of osteosarcoma. There's also some other genomic study early, for example, from St. Jude. They also did this analysis. So basically what we found in this study is we have included about 120 patients, and we did multi-omics study, and we trying to subtype this osteosarcoma. So this is a landscape profile. The finding is basically the same as previously published. There's like TP53 mutation is common, but also there's a very high frequency of MYC. 8q amplification is very high. Well, we were able to, by the multi-omic analysis, we were able to show that the MYC amplification is the main prognostic factor leading a very, very poor outcome. These patients are, they progress very, very quickly with multiple metastasis to the lung, to the bone. So we categorized into 4 subtypes. Subtype 1 is immune active subtype, which this patient has the best outcome and the best immune infiltration in the TMA. These patients has relatively stable and less mutation, have a stable genomic. Subtype 2 is, or we name it immune suppressed subtype. And these patients have less inflammatory or less activated TMA. However, we were able to show that there's still some functional T cells in the TMA. The outcome is a little bit poorer, but like 60% of 5-year overall survival. Subtype 3 is the HRD dominant subtype that has a genomic scar phenotype and a very high genomic instability. The outcome is relatively poorer, but we hypothesize because maybe it's relatively sensitive to chemo agent like cisplatin, so this the outcome is a little bit poorer. The subtype 4 is the most has the most progressive phenotype. We name it MYC-Driven. So let's see the next slide. Okay. So the MYC-Driven type has this gene expression profile of highly metabolic phenotype, and has very high, also very high genomic instability. but it has very low immune infiltration and has the poorest outcome. Okay. And also, this is the immune landscape of the different subtypes. Subtype 1 and 2 is has immune marker expression. However, the subtype 3 and 4 is like the immune desert. There's no desert. There's no T cell or other immunocells in the TMA. Okay, so we believe that most of the patients we see in the clinic with relapse disease is the MYC-driven type. So that's why when we do clinical trials, there's a very high rate of failure because this group is not sensitive to, it has no immune infiltration. So earlier, there's a lot of immune checkpoint inhibitor clinical trials for osteosarcoma, but all of them failed. And the response rate of PD-1 for osteosarcoma is so low. So this figure is a representative patient with very high MYC amplification and very progressive long metastatic disease. And the immune score we can see is very, very low for MYC subtype. And we are now doing a proteomic study to further validate this subtyping system. So the work is almost completed. We're now doing some validation using slides and some other. We found the same. We found also in proteomic study, we confirmed that MYC is the most strong prognostic factor. MYC amplification leads to very poor outcome, and also very high metastatic rate, very low, very low chemoresponsive necrosis, low immune infiltration, and the tumor is highly metabolic, highly proliferative, and also the downstream target of MYC protein is highly expressed. We also find some downstream target. Now we are doing some validation to see if it can be prognostic or it can be therapeutic targets. And we did a very pilot study trying to suppress MYC expression and to see if it can be therapeutic. This is the umbrella trial for subtype 2 and 3 and 4. So this design is to select patient using either NGS or immunohistochemistry, and we divided them into the immune therapy group and the HRD group using PARP inhibitor and DNA damaging agent. And in the MYC-driven group, we use this drug, tinegotinib. This is a drug from Chinese pharmaceutical company. It's been in clinical trial for now. It's in breast cancer and some other cancers, and I think it has also been clinical trials in the MD Anderson or some place else. but we did it in a very small number of patients in osteosarcoma type. Before clinical trials, we firstly did some lab work to test it can inhibit MYC. It can. I think it's sorry. There's some other things. This is a aurora kinase inhibitor, and we know that aurora kinase can stabilize the MYC protein expression. So as we can see, it inhibits the phosphorylation of aurora A and B, and the aurora A and B can stabilize MYC. So when the phosphorylation is inhibited, the MYC protein can be degraded. That's the basic mechanism of this drug. So we did some animal study to see it's able to reduce the growth of the tumor, and also we have shown that when we able to inhibit MYC, we can upgrade the immune therapeutic effect of like PD-1. It's a synergistic effect of these two drugs. So we did this pilot study in osteosarcoma. However, most of the patients, the best outcome is stable disease. There's some regression, but cannot last very long. There's only one patient. We have in subtype 2, which we use PD-1. It showed very good outcome. The patient has a PFS about 1 year, and some of the tumors shrinked. In MYC-driven type, however, there's tumor necrosis, but it cannot last very long. So the overall PFS is progression survival is not very long, like 3 months. 3 months. We have the patient enrolled in this trial is most of them are very late stage, 3-4 lines of therapy. So, and also we think that the single drug treatment of aurora kinase inhibitor or other MYC inhibitor cannot effectively make the tumor regression. There's also other MYC target drug like the MYC degrader or PROTAC, and MYC-MAX, mimic peptide something. There's one paper in published last year. The drug is OM0103. The result is almost the same. The best outcome is SD. There's also other study.
Right, Dr. Hua, can I interrupt you? There was just one question, clarifying question that came in. What measure was used for the c-MYC IHC positive negative threshold?
Oh, threshold. The c-MYC, the amplification, most of them is very high, like 8, for example, 8, 10. This from the NGS copy number variation. Most of them is very high. So The threshold we use is 8. Is 8. So because all the patients enrolled, this is very very high MYC amplification. So the very progressive disease. But there's also several patients. They have their NGS from patients enrolled all over the country. They have a genomic report from other places. We did not have this raw data for his NGS, and we use IHC to it reports that they have a MYC amplification. So, but we validate its highly expressing in IHC, like plus, 3+. There's also like 2 or 3 patients we use. We didn't do NGS by ourselves. Yeah, we can discuss later.
Thanks for answering that.
Okay. There's also other studies. We're trying to study other subtypes. For example, there's also very high frequency of 9p loss. Within this region, the MTAP is lost, is deletion. It related also to immune suppressive TMA. In this work, we are able to show that when in this subtype, MTAP deletion related to immune suppressive TME and methionine intervention can also induce this PD-L1 expression and enhance the immune checkpoint inhibitor therapy by the MAT2A inhibitor, which is can also have this methionine intervention effect. And we were able to see that in this group of patients, there's some of the patient with the MTAP deletion can benefit from the PD-L1 treatment. This has also been published. And so the next thing we are focusing is the extrachromosomal DNA in osteosarcoma. We believe that this is the outcome from Chromothripsis. This shattered DNA chromosome has this created this ecDNA. It's outside chromosome, and it's very specific in cancer. It's not in the normal tissue, and the oncogenes like MYC, EGFR, something else is amplified in this small circular DNA, and very high transcriptional activity. And because it's outside of the genome, so it's like randomly divided during mitosis. So it's non-Mendelian inherited during the division. For example, we can show here there's when the cell divided, some of them acquired more ecDNA, some of them is less. And during the drug treatment, these cells with less ecDNA, they are more sensitive to chemotherapy, to radiation, and the cells have acquired more ecDNA, has higher MYC amplification, survived during the treatment, and leading to eventually drug resistance, and we can also see this in the clinic, like multiple relapse. After multiple relapse, the tumor become more and more aggressive. So we tested, this is a paper, this is a result from a published paper. The ecDNA is originated from Chromothripsis and induced genomic fragmentation and reassembly. The sarcoma has very high expression of this ecDNA. And this is a pan-cancer study of like different different types of cancer from the UK genomic project, 100,000 genome project from the Great Britain, studying ecDNA, and it showed ecDNA is associated with poor prognosis, very higher stage of disease. And in this report, they reported about 50% ecDNA positive in osteosarcoma, which is also consistent with the Chromothripsis study in cell. This figure showed very high rate of ecDNA in bone and soft tissue sarcoma, especially osteosarcoma and liposarcoma. And there's also some study trying to target this event, to target ecDNA positive cancer. One of them is the checkpoint 1 inhibition, the CHK1. This is a transcription, the checkpoint during transcription, and they were able to show that ecDNA exhibit high transcription replica conflict, so it's vulnerable to CHK inhibition. This CHK inhibitor selectively kills ecDNA continuous cells and prevent drug resistance in the clinical study. There's still some key questions we need to answer. For example, what is the ecDNA landscapes, and what is the downstream event of ecDNA positive? Can this be a diagnostic tools, a biomarker for osteosarcoma subtype, and how does it play in the progression, heterogeneity, and drug resistance? We still need to have this some described study for osteosarcoma, and most importantly, can this be targeted? Can this be therapeutic? And what experimental model can we use to further study ecDNA in osteosarcoma? So we are able to study this. We grabbed the published data, and we combined our own data with in our Shanghai General Hospital osteosarcoma cohort. Currently, we have about 240 samples from 200 patients. We created a bunch of osteosarcoma PDX models. And this is some preliminary work from the cohort. Our result is the same as reported, that half of osteosarcoma patient harbors ecDNA, and the most well-known oncogene we know that is amplified mostly very commonly in ecDNA. not in the genome. And ecDNA positive is related to very poor prognosis. So this is the patient we have. And we can see this is outcome is the outcome is very significant, and there's a lot of well-known oncogene, for example, cyclin E and MYC, CDK4. They are all amplified in the ecDNA positive patients, and very high level of MYC amplification in the forms of ecDNA. The oncogene amplification on ecDNA is significantly higher than other forms of amplification. This figure you can see. So the target, the oncogene MYC, we hypothesize this leads to very poor prognosis, and they are amplified in ecDNA. So that's why we are trying to we are focusing more and more research on ecDNA studies. And also, there's also some these immune suppressive genes, which is also amplified in ecDNA. We did this transcriptomic study showed that ecDNA positive tumors exhibit suppressive immune pathways and activated proliferative signaling. For example, this NOC1 and CNPV, CNPV and the TNF family members, and the IL-22. These are all immune related signaling, and we were able to show that the ecDNA positive cells compared to ecDNA negative cells, the immune pathways is suppressive. And also we did this in our PDX model. Interestingly, in PDX model, maybe it's during the passage, the ecDNA somewhat rendered the growth advantage. So in PDX model, there's even higher incidence of ecDNA positive sample. The copy number variation is also higher in the PDX model, and most aplicounts are consistent between PDX and human tumor. These are the PDX model we created. We can see these are all Chromothripsis, Chromothriptic tumor, and there's a very high copy number of variation in PDX. And maybe there's a clone selection during the PDX passage. So the ecDNA positive clones display growth advantage and more aggressive phenotype in animal model. So that's I think the experimental model we can use in the further study. And also ecDNA amplification is also heterogenetic. For example, in this patient, heterogeneity within the tumor. For example, this in this slide we can show here this region is a higher copy number of CDK4 in ecDNA, and this region is less. So which again has somewhat indicate us as the variation between ecDNA distribution in within the tumor may leads to a heterogeneity during the treatment, and also during the treatment, it can the ecDNA positive cells they gain the growth advantage and can be accumulated during therapy, and eventually leads to a high ecDNA enrichment, and leads to the chemotherapy resistance. So these are very pilot studies. We are also trying to expand our cohort and trying to characterize ecDNA difference between the different subtype, and trying to test if there's some drug that can target ecDNA positive tumor compared to ecDNA negative tumor. And the most important thing, well, how we can diagnose this and how we can have this prevention in the ecDNA evolving. We do not want to our treatment we do not wish our treatment is a process of enrichment of the ecDNA positive MYC-amplified subtype, and eventually we can do nothing of the late stage patient. So the early intervention of surgery of metastatic lesion to resect the lung mets is also important for a physician decision. So this is a database we have right now. We have about 300 patients in our biobank. We have all these data available. We're still now we have this government-funded project. Not only our hospital, we will combine the 4 main hospital in Shanghai to create a bigger cohort called Shanghai Bone Tumor Cohort. There's more funding for us to do the multi-omics study in the future, in the future 3 years. And this is our PDX platform. We have more than 150 PDX. Half of them is osteosarcoma. We have also some other cancer type like chondrosarcoma, leiomyosarcoma, Ewing sarcoma, synovial, what is PS? Can't remember. Epithelial sarcoma. And we have profiled all these PDX models. And my PhD student, Wei Song, he is also online. He's grabbing this data together, describing all the phenotypes, but he is mostly interested in ecDNA. So in this study, I think we are going to submit this year. We will describe ecDNA profile in these PDX models. And this is a genomic study for all these PDX models. The findings are basically the same as in human samples, not very different from what we know in the past. And also, the last slide I want to show is we created some of the organoid models for osteosarcoma, and we're using this to test the drugs, different preclinical agents, compounds, and also we integrated some of the immune infiltration and some of the vascularization in the organoid model to mimic the how we can mimic the immune environment in vitro. So this is a very very brief introduction of our work. I think a lot of things needs to be done, and I'm very glad that we have this group, scientific group of MIB factor focusing on osteosarcoma. Thank you very much.
Thank you, Dr. Hua. That was a lot to get through, a lot of work. Everyone, please feel free to put any questions in the Q&A if you have any questions for Dr. Hua. And maybe I'll kick it off with a few on just some questions on
I stop.
Oh, sure. You can stop sharing.
Yes.
Yeah.
Okay.
There you go. For the umbrella trial, so you did mention that many of the patients on the umbrella trial were heavily pretreated with about 3 to 4 lines of therapy, and I was curious, was bulky disease resected at that point? And because I know sometimes when patients enroll in a trial like that, it's almost like the agents don't have much of a fair chance because they're at such an advanced. So,
sorry, for the patients?
Yeah. So for the patients that were enrolled in the umbrella trial, you mentioned that they had undergone several lines of previous therapy. And I was just kind of curious what disease stage, if bulky disease had been resected, just because, again, there's at such an advanced disease stage, sometimes it's hard to kind of give the agents that you're testing a chance to actually do anything. But just curious kind of what next steps are after that umbrella trial, and if there's a way that you could potentially have the molecular subtyping kind of guide upfront therapy choices versus having it come kind of later in a relapse setting.
Yes. Most of the patients is highly metastatic, lung metastatic patient. So for patients treating in our hospital, we do the NGS beforehand, a little bit earlier. For example, after their primary surgery, primary tumor surgery. When people from all over the country coming to have this enrollment, they have their slides, but it mostly take 1 month to get the result. So the disease are a little bit late. The problem we are trying to figure that whether we can do molecular subtyping in biopsies, but the sample is very small. We still need to keep most of them for pathologists to have a accurate diagnosis. So the material we can use is very limited. So that's the problem we are trying to figure. It's always better to push everything beforehand.
Yes, I see. So you are limited by the tissue you can get from the biopsy. So maybe if the testing can become more sensitive, or you just need less tissue volume, it'd be easier to push it.
I think yes. I see questions in the chat box.
Do you want to take?
Yes, decalcified tumor for molecular phenotype. Yes. Decalcified perfume embedded tissue for genomic sequencing is no problem for that, but it's not good for using decalcified tumor to do the IHC validation. And also, most of the time we use fresh tissue, but if the patient come from other institute carrying his slides, we can do the NGS. Yeah, no problem. But only the WES, mainly WES. It's hard to do the RNA. The RNA-seq, yeah, now we can do RNA-seq using the PFFE, yeah. I'm sorry, I can hear your...
Oh, so sorry. I just a couple questions about the ecDNA, which I thought it was so interesting. I think you had a slide that I'd seen that where it was kind of like it's, imagining the Chromothripsis, and this is basically kind of almost like some of the debris, from the Chromothripsis. And so there's kind of that correlation between Chromothripsis and ecDNA. And but I guess what I wasn't sure, so you're talking about CHEK1 inhibitor potentially as a way to target ecDNA. And then you had mentioned that that ecDNA has oncogene amplification, like higher almost amplification of these oncogenes.
Yes.
So if you were to target those oncogenes with, some sort of inhibitor, would that also affect the ecDNA or no, because it's outside the chromosome? Do you understand?
Yes. It's not directly. It's not directly targeting these oncogenes in ecDNA. It's just a replica. The higher it has for cells has very high MYC amplification. It has very high also has very high rate of replicant stress, meaning during the mitosis, the cells are vulnerable. So the checkpoint one, CHK1 is functions during the mitosis, and during this step, the CHK inhibitor can causing more replica stress and to kill the cells with high MYC amplification. It's like a synthetic lethal mechanism. But also, I've discussed this with some with the industry. The CHK1 inhibitor is very the toxicity is very concerning problem. So not very in pharmaceutical industries interested in this target. Only a few of them are doing the drug development.
But you mentioned
we still need to find some other ways, better target.
Yeah, but you mentioned that there is, high levels of ecDNA in other cancer types, right? With
other cancer types, yes.
Potentially more patients that hopefully might drive more investigation or investment in CHEK1 inhibitors.
Yes. In small cell lung cancer, in the late stage breast cancer, also it's a very universal. I think it's a problem for most of the copy number variation prominent cancer. Yes.
Yes. There are a couple of questions that came in, so let's get to those. Have you observed a lot of heterogeneity in genes amplified in ecDNA across patients? Can you expand a bit more on that? And what do you think is the most promising therapeutic strategy to target genes amplified in ecDNA?
A lot of gene heterogeneity in gene amplified. The gene amplified in ecDNA, yes, it's not all the same. It's not all the same, but there's a lot of genes that we are not. There's a lot of genes in amplified, but we're not very familiar with the function. So the thing come up first is those oncogenes that we are well-known oncogenes like EGFR, also MDM2, MYC, cyclin E. So in this work, we are trying to profile more genes. I think Wei Song can maybe describe a little bit more. Can they talk, the audience?
They can't. Let me see if I can figure out a way to do that.
Yeah. If not, I think Wei Song can type. Yes, it's no problem.
Oh, there. I think I actually just gave him permission. He wants to jump in.
Yeah, Wei Song, can you describe more about the gene distribution in the ecDNA panel?
Can you hear me?
Yes.
Yes.
Yes. A lot of genes amplified on ecDNA. Sorry, I type later.
Okay. Yeah.
Thank you.
You can type your what we found in the chat box. What are the consequences of non-canonical TP53 structure variation in ecDNA? The consequence in ecDNA. Yeah, I'm not sure how to answer this question. Yeah, we did not go through the TP53 different type of variation in structure variation in P53. We have not looked into this particular. Sorry. Yeah.
Okay, before we let you go, Dr. Hua, I think we have time for one more question. And since we don't have any in the Q&A, I might just ask you, so it's the Shanghai Bone Precision Platform, the 4 hospitals that you talked about. That's fantastic that you're doing that. And kind of wondering how and where do you collect your data, and how might that be accessible to other researchers? Because you have such an incredible data set, with the 200 plus samples. And it sounds like some of those, so it sounded like you had, it was like 240 something samples, 200 patients. So some of that might be longitudinal data in the same patient from primary to relapse or something?
Yes. We have uploaded part of the data into a Chinese genome bank in Beijing, and the link, I think I can send you the link later. But this was only part of the published data. The data we are trying to collect in the next 3 years, we will also have a way to share to the community where from Shanghai. There's Shanghai government, they will create another link or a website or something else in the future. I'll let you know.
Yeah, that would be great if you could share that, because I'm sure that would be helpful. In the same way that I think it's so great that you're pulling information from all the other sources, like the St. Jude and Target and all those things. It would probably really enrich a lot of people's work to have access to that.
Yes, we want to do a re-analysis, combined data set.
Fantastic. Well, thank you so much, Dr. Hua, for joining us on OsteoBites today and for making it better for pediatric osteosarcoma patients. More information on this and all OsteoBites can be found on our website at mibagents.org, on the MIB Agents YouTube channel, and at your favorite podcast place. And we will be back with OsteoBites on July 10th. We're taking a bit of a break in June. We're going to be joined on July 10th by Dr. Kelly Folk. She's a pediatric oncologist at Children's Hospital, Colorado, and she will review an open phase 1B clinical trial evaluating the combination of losartan and sunitinib in the treatment of pediatric and adult patients with relapsed or refractory osteosarcoma. You can find our OsteoBites lineup for the next few months on our website, and if you have ever any topics that you'd like to hear about, please feel free to email us at events@mibagents.org. Thank you again, Dr. Hua, for spending time with us today, especially at such an early hour for you. And thanks to all of you for taking time out of your day to join us on OsteoBites. We hope to see you back here soon in a month, a little bit more than a month, on July 10th. And in the meantime, we hope to see you in person at Factor in Salt Lake City. Thanks so much. Thanks so much, Dr. Hua. Thanks, everybody.
Thank you, everyone. Wei Song has typed some of the note for ecDNA. It's great to have discussed with you, and we will see you soon.
Thanks so much. And I just popped Wei Song's notes in the chat if anyone wants to take a look at that. All right. Thank you so much. Thanks for having a good rest of your day. Thank you.
Bye-bye.